So basically, the whole idea is that we're going to talk about phospholipids, and because they comprise an important, basically the main part of biological membranes. Okay, so we're going to chat about the main components and the properties. So there are two types of phospholipids that can be found in the membrane of cells: sphingomyelin and glycerophospholipids. So glycerophospholipids, as you can see, they contain four principal components. The glycerol residue, that's a polyatomic alcohol, two fatty acids. We're going to talk about some amino alcohols, but really important, the phosphate group. So this phosphate group essentially forms two ester bonds. So this and this are ester bonds because, remember, ester is formed between an alcohol and the acid. Well, phosphate residue is basically a residue of an acid, the phosphoric acid, while amino alcohol and glycerol are, well, alcohols. So there are three different amino alcohols that are found in glycerophospholipids: choline, serine, and ethanolamine. Why are they called amino alcohols? Because they have an amino group and they have hydroxyl groups. These hydroxyl groups, they will form an ester. Okay? Does that make sense? Now, amino group NH2 is a really, really strong base. So at the physiological pH of 7.4, this amino group works as the Bronsted Lowry base, which means it gets protonated. It accepts the proton. So far, making sense? So, hold on a second. Give me 1 sec. I'll explain. I'll just illustrate it for you right now, okay? So, for instance, we have this acid H3PO4, right? So, I'm going to do the structural formula. So this is phosphoric acid. Does that make sense? So you add the OH group, the alcohol part. So what you end up with is this. Clear? And water goes away. Am I making sense right now? Good? Okay. So a couple of examples of glycerophospholipids: lecithin and cephalin. Okay? Pretty abundant in brain and nerve tissues. Okay? The difference between the lecithin and cephalin, you can see that fatty acids are the same, but the amino alcohols are different. Okay? Am I making sense? So this is probably the most important idea that we're going to discuss regarding glycerophospholipids. That they contain both polar and non-polar regions. So there's a polar region that can respectively interact with polar substances such as water. And then there is a non-polar region that interacts with non-polar substances. So the polar part of glycerophospholipid molecule is usually termed a polar head. So what is it comprised of? It contains an amino alcohol, like a choline residue, and a phosphate group. Okay? If you think about the structure of the amino alcohol, they really kind of a small molecule. Can you see that? They're not huge. They're like what? 5 carbons in choline, 3 carbons in serine, and 2 carbons in ethynyl amide. So the fact that they have charged groups here, and they have highly polar OH group, all of it explains why amino alcohol is a polar residue. Okay, does that make sense? On top of that, you have a phosphate portion. So phosphate portion, if we are absolutely correct about how it looks, I want you to take a look at this structure. Take a look at this part. Okay? And I'm going to illustrate something here. Okay, so this is the phosphate group. It forms 2 esters. One with amino alcohol. Everybody agrees? And then another one, I'm going to turn it to, sorry, I just did something horrible. Okay. So, and then another part of it would be glycerol. Agreed? Basically, I redraw what was at the title page. But we have a couple more bells and whistles hanging off of the phosphate. At physiological pH, phosphate is charged, okay, which makes it even more polar. Am I making sense at this particular moment? Good? Okay. Now, the nonpolar part is that hydrocarbon tail, which is essentially fatty acids, okay? Basically, the structure of glycerophospholipid allows it to form membranes that would regulate the transport of substances in and out of the cell. So, essentially this portion, the fatty acids plus glycerol, okay, so fatty acids plus glycerol is nonpolar. That portion here, amino alcohol and phosphate, is essentially polar. So far we good? Everybody? Okay. So why some illustrations of why knowledge like this is important. The venom of some snakes contains the enzyme called phospholipase. Phospholipase can break down phospholipids, essentially destroying cellular membranes. And that may, for instance, result in hemorrhages, bleedings. And since we know the structure, we know how it works. So another type of phospholipids is sphingomyelin. So the reason why previous group was called glycerophospholipids is because they contained glycerol. This group of molecules doesn't. Instead, it contains sphingosine. There's only one fatty acid residue also. So how does it all look like? So there is an amino group in the molecule of sphingosine, which forms not an ester, but an amide with a fatty acid. Sphingomyelin also contains hydroxyl group, which forms an ester with phosphate, just like in the glycerophospholipids. The amino alcohols that can be present are either choline or ethanol amide. Sphingomyelins found in myelin sheath. The coverage that surrounds many of human neurons, not all of them, but many, the majority of them, providing electrical insulation and increasing the rate of transmission of nerve signals. So this is the structure of sphingomyelin, and let's take a closer look at this. I'm going to highlight different parts of it. So first of all, we need to identify the sphingosine molecule. So this is the sphingosine. Make sense, everybody? This is fatty acid. This is the phosphate part. And this is amino alcohol. Am I making sense right now? Now, I'm going to give you that list here so you can, kind of orient yourself. So the yellow is sphingosine. The pink is fatty acid. The blue is phosphate. And the green is an amino alcohol. So far so good. Now, let me roll back to glycerophospholipids here. Yes, this part. So this bond between the glycerol and fatty acid is an ester bond. Sounds good so far? When you look at sphingomyelin, this bond is an amide bond. Making sense? All good? You can see there is a small difference. Instead of OH at this carbon, it's NH. So now we're going to talk about steroids, cholesterol, bile salts. I'm going to highlight some of the misconceptions regarding these. But let's start with the definition of a steroid. Steroid is derived from this nucleus, okay, that contains 4 condensed rings. 3 of them, as you can see, are 6-member rings. And 1 is 5-member ring. Okay? You don't need to know the numbering. There won't be questions like number the carbons in those rings, okay? But the point is that in cholesterol, there are 2 methyl groups attached to those rings. So cholesterol is true, it is the most important and abundant steroid. It has a hydroxyl group, a double bond, 2 methyl groups, and a fairly long alkyl chain at the 17th carbon. Okay? So let's do some myth busting, shall we? The first statement, cholesterol is obtained from meats, milk, and eggs. I would replace is with can be. It can be obtained because it is in eggs, in milk, in meats, in virtually any animal product. Do you follow my train of thought? About 15% of the cholesterol that your body uses every day come from food. 85% is synthesized in the body. So, myth number 1, if you're going to eat less eggs, your cholesterol is going to be low. No, it won't. Myth number 2, you're going to eat like 2 eggs a day, you're going to die of a heart attack. No, you're not going to die of a heart attack. It's actually not going to affect the cholesterol levels in your body. You can eat 6 and it's going to be totally fine. There are genetic conditions associated with cholesterol metabolism. I'm talking about physiologically healthy person, all right? Sounds good? Okay. It is essential for cell membranes. And this. No. The mechanism is a little bit more complicated. It says high levels form plaque. So basically what it suggests is that when you have high cholesterol, right, just high cholesterol, it kind of floats, like chunks of it float in your blood, and they for some reason get stuck to the wall of the vessel and eventually clog it. This is not how it works. If we will have time, I will briefly mention, just explain so you feel better about your diet, okay? So cholesterol is considered elevated, yes, if the plasma cholesterol exceeds 200 milligrams per deciliter. So here's the thing. Multiple studies demonstrated that total cholesterol levels in the blood cannot serve as a predictor of cardiovascular events. In other words, if your blood cholesterol is in the thousands, something is horribly wrong. If your total cholesterol is consistently 220, 240, it says nothing. It doesn't say that you're healthy. It doesn't say that you're not. It just says nothing. We will get back to this later. Okay, sounds good? So, this is the very careful statement. So yes, it is true that the diet that is low in saturated fats appears to lower cholesterol levels. But again, is it associated with a decreased risk for cardiovascular disease? And the short answer is not really. So basically my point, if you like eggs or milk or butter or beef, go for it. I mean, obviously, if you decide that your diet will consist of 4 pounds of steak and 2 butter sticks, and you're going to wash it down with a gallon of milk every day, that's probably going to affect your cholesterol metabolism overall because of the enormous amount of fat that you will have to process. But the regular healthy garden variety diet that incorporates meat and milk and dairy and whatever, it's fine. Okay? So cholesterol in foods, as we mentioned before, is a fairly useless information because only about 15% of your cholesterol intake comes from food, right? Does that make sense? Only 15% of cholesterol in your body is actually from food. 85% are synthesized. I mean, you have to put some really enormous effort to change your circulating cholesterol levels through food. Like, I don't know. I mean, nobody did that deliberately, but I would guess like a dozen eggs a day plus half a beef liver and top it with, I don't know, a load of milk and butter, then yeah, probably. But if you eat regular diet, no. Okay, sounds good? So actually, let's talk a little bit more about cholesterol. Have you heard about high density and low density lipoproteins or good cholesterol and bad cholesterol? Okay? So there's HDL, which is good cholesterol. You want it to be up. And there's LDL and VLDL. You want it to be down. Let me explain why. Okay? So remember how the statements said that, cholesterol can block your arteries, right? So here's what happens. There are cells in your blood vessels that can the process is called scavenging, okay? It can uptake cholesterol. Am I making sense? They take it in. If they take in bad cholesterol, your vessels become inflamed. In response to that inflammation, a bunch of white blood cells get stuck around the area that is inflamed. These white blood cells will capture cholesterol, yes, and they will incorporate it into a growing plaque. Is it clear? Does that make sense? So it's mostly your immune system that does it, not circulating cholesterol levels. And two people can have same levels of cholesterol, but the ratio between high and low density, ratio between the good and bad, can be different in those people. And it was shown, hear me out. So I think it's the opposite. So what you need to look at is LDL to HDL ratio. You want it to be small. Am I making sense? You want a lot of HDL, not a lot of LDL. This is the ratio that you're looking for because total cholesterol measures both of them and it's not informative. Sounds good? Okay, now bile salts. Bile salts they help in absorption of all fats, actually, not just cholesterol. Okay? So this is what a bile salt, this is what it looks like. So this part, basically your steroid nucleus, the steroid part, is nonpolar, right? And this part, you can see there are charges, there is nitrogen, 2 oxygens, all of it makes this, the glycine residue, highly polar. Are we good? Okay, so you have, we call these molecules like phospholipids, and bile salts are amphiphilic, or sometimes they're called amphipathic. They contain both polar and nonpolar. Making sense? Now, why do you want this? Why do you want polar and nonpolar? So imagine you have a greasy skillet, okay? And you want to clean it of the fat, so you will add dish soap, right? Dish soap has molecules that compose dish soap, have a polar part that interacts with water, a nonpolar part that encloses the fatty molecules and make them soluble. So if you represent your bile salt like this, okay, where the circle means the polar head and the stick means nonpolar tail, okay, it will be able to solubilize fat in your intestines, any fat, steroids, fatty acids, doesn't matter, and help with uptake into the blood. Am I making sense? All good? So, couple of things here. First of all, so bile obviously is required for fat absorption. People who had cholecystectomy, removal of the gallbladder, what kind of food should they be careful with? They don't store. They still have liver. Liver will still make it. They just can't store bile in the gallbladder. So they should be careful around what type of foods. Fat. Yeah. They can eat it. It just has to be like very well paced. You can't just, again, eat a stick of butter. Okay, you're going to be very, very sick. Am I making sense right now? Second, yes, it is true, the gallstones can be formed. Gallstones are formed when the amount of cholesterol, bile itself contains cholesterol. Am I making sense? It has cholesterol in it. It does. When there is excess of cholesterol in the bile, when you have real hypercholesterolemia and bile has to remove it, you can form gallstones. They're not stones, they're actually pretty soft. But this is my favorite question I ask all the students. You're familiar with the concept of gallstones, right? What is the largest gallstone ever removed from a human so that this human lived afterwards? Mass. I'm asking about mass. Quarter of a pound. Any other ideas? I'm going to give you 3 guesses. Any other ideas except quarter of a pound, more or less? What do you think? What do you think? More or less? The right answer. 14. 14 pounds. The largest gallstone ever removed, extremely obese British woman. It was the size of a watermelon. Obviously, they removed a gallbladder, but yeah, she lived. Okay? That's pretty impressive. Okay, so now we're going to chat a little about the transport of lipids. Okay? So cholesterol is esterified in the blood. This is essentially fatty acid residue that is forming an ester bond with a hydroxyl group on the cholesterol. Okay? Now, I want you to take a look at this molecule and tell me one simple thing. Overall, based on your knowledge of organic compounds, is this molecule polar or not? What do you think? Huh? Non-polar. Of course not. Fatty acid. Non-polar, right? This whole cholesterol thing. Non-polar. So it's conceivable that this is non-polar. Now, what is the main component of your blood? Say again? Yes, it's water. Okay. Is water polar or not? It's polar, which means non-polar molecules, you cannot transport them in the blood. So you have to somehow, kind of make them polar. So this is the idea. These non-polar compounds, such as fatty acids and monoglycerides, whatever, cholesterol, okay, they are transported in the form of lipoproteins, which is a pretty complicated structure, okay? So as a core, you have non-polar lipids, okay? Which is the esters of cholesterol esters, we just saw them, triacylglycerols, super nonpolar compounds. And they are surrounded on the surface by proteins, which are polar, phospholipids, which have a polar part that sticks out, so there's polar heads, and cholesterol, which is incorporated there, okay? So basically, you have a two-phased structure. The surface is polar, and the core is nonpolar. Okay? Now, so imagine you have butter, liquid butter, right? Or bacon grease. And you pour it into a glass of water. Where will fat be? Will it sink or will it float? Float. It will float because the density of fat is lower than the density of water. Does it make sense? Now, the simplest example, let's say you have a piece of beef and you throw it in water. What's going to happen to it? Sink or float? Sink. Because the density of a protein is more than the density of water. Am I clear? So now if you look at, we're going to skip chylomicrons. I just don't want to talk about them. When you look at VLDL and LDL, okay, this shows you the density. You can see that the density of VLDL and LDL is lower than that of HDL. Why? I'm going to highlight this bottom part. Look at the percentage of proteins within each complex. Remember, proteins have much higher density than lipids. Do you understand what I'm trying to say? So if you have very small amount of protein, the overall density is low. This is why we call it very low density lipoproteins. But then as the amount of protein increases, the density increases. And this is why we call it high density. And the thing is, if you look at this specific image, you see the direction in which low and also very low density lipoproteins transport fats. They transport from liver to the tissues. Can you see that? Can you acknowledge that? What do you think happens to fats in your, say, muscle or your heart? What is it used for? Fat. Source of what? Say it. Source of energy, yeah. Okay, does that make sense? Now, we go back to this little conversation here. HDL returns from tissues back to the liver. Am I making sense? So, these two are to the tissues. HDL from the tissues. Cool? Now think about this. You send a bunch of molecules that contain a lot of fat, and then your tissue will use that fat and return unused stuff back to the liver. So the more fat your tissues can use as a source of energy, the less HDL, the less low density lipoproteins you're going to have because they're all going to be used, and the more high density lipoproteins you're going to have. Am I making sense? Well, how can you increase the energy consumption by your muscles? Yep, exercise. And it doesn't matter. So first of all, it's been shown that for cholesterol control, resistance training is much better than endurance exercise. Okay? But, like seriously, just walking around, going for a jog, like doing some fairly simple exercises will go a long way in controlling your blood cholesterol. Now, steroid hormones. So we have sex hormones and some of the hormones produced in the adrenal cortex. So basically the idea, they regulate various body activities and they are produced from the cholesterol. Okay? So in males, for the most part, it's testosterone. Okay? Androsterone plays very secondary role. In females, it's estrogen. So estrogen and testosterone play very similar roles in females and males, respectively. Progesterone is kind of unique because the main role of progesterone is to assist the fertilized egg to implant into the endometrium of the uterus and kind of facilitate the development of the fetus. Does that make sense to you? Estrogen and testosterone have slightly different function. And then we have corticosteroids that are produced in the adrenal cortex. Adrenal gland is right on top of your kidneys. So there are mineralocorticoids, basically aldosterone that regulates the water and sodium balance, and glucocorticoids, which are the hormones of the stress response. They essentially increase your blood glucose. Okay? So these are some examples of steroid hormones. So testosterone produced in testes, estrogen produced in ovaries, just like progesterone. Okay? And then this, norethindrone, synthetic progestin, which is an oral contraceptive. Okay? But this is the interesting part. I have somewhat unconventional view about that. Travel voice. So basically, testosterone facilitates growth of muscles. And it was used by bodybuilders for a pretty long time. Okay? Sounds good? To facilitate muscle growth. There's only one problem. Well, not one. There are several problems. One, if somebody takes testosterone, injects testosterone, it's going to, because of the mechanism that we don't have time to go over, it will significantly reduce testosterone levels in your body and eventually results in infertility. The testes will shrink. Another problem is that testosterone is metabolized into estrogen by the enzyme called aromatase in the liver. Not only it damages the liver, excessive levels of testosterone, it also results in the imbalance eventually and accumulation, paradoxically, accumulation of estrogen, which leads to the development of secondary sex characteristics. So the quest was, can we make it sort of safer? The answer is yeah. So various derivatives of testosterone that have much less side effects than testosterone itself. Still going to be liver damage. Fluid retention is a big part of it, yes. It's true. Because they steroids. Hair growth, I don't know if it's a negative thing. Sleep disturbances and rage in general, yes. Okay? But this is the statement, they use it illegally to increase muscle mass. I personally think, and finally some adrenal corticosteroids. So on top of each kidney there is an adrenal gland. It looks like a little pyramid. And its outer layer called cortex produces aldosterone, which fine-tunes the electrolyte balance, basically sodium and potassium balance, and cortisone, or cortisol, which regulates glucose. So this statement is plainly wrong. You can see that those were chemists that wrote it. No, it doesn't stimulate glycogen synthesis. It actually breaks down glycogen in the liver. Cortisol indeed increases blood glucose. That is true. Okay? So these are adrenal corticosteroids. So these two are slightly different forms of cortisol and cortisol. Okay? Those are chronic stress hormones. And I want to highlight chronic stress. Am I making sense? Not you being cut off on the highway stress, but stress of, I'm waiting for my immigration status confirmed and USCIS does not send me the papers and I'm in this limbo for like last 3 months. And I don't know what's going to happen. That's a chronic stress. Make sense? Aldosterone. Now, this is prednisone. So very similar to cortisone, basically used. Both types of glucocorticoids are strongly immunosuppressive. So prednisone is a pretty strong immune suppressor. And these are adrenal glands on top of the kidneys. Does that make sense? Now, it brings us to the conversation about the cell membrane organization. So I'm going to skip that introductory slide. And the idea is that cell membrane separates the content inside of the cell from the outside. And all membranes in animals, plants, fungi, and most bacteria consist of a lipid bilayer. Okay? So, phospholipid bilayer. Remember that phospholipids have the polar head and non-polar tail. So polar heads are facing either into the cell or into the environment, and hydrophobic tails form the middle of the membrane. So here's how your membrane looks like. You can see that the green, the heads, are facing either inside or outside of the cell. Why? Because the content of your cell and the extracellular environment are mostly water. So it's polar. So you want a polar head to face a polar solvent. Is it clear? However, the inner portion is made of fatty acid tails, so it's non-polar. Okay? Am I making sense? All good? And other two large components of the membrane are proteins and cholesterol. Okay? So I have essentially three types of molecules: phospholipids, proteins, and cholesterol. And the membrane essentially behaves more like a fluid, but a very viscous fluid. Am I making sense? It means that the molecules within the membrane can actually move. Proteins can move, phospholipids can move, cholesterol molecules can move laterally through the membrane. Okay? Now, if you remember the story about saturated versus unsaturated fatty acids, right? Saturated fatty acids look like this. So they tend to stack pretty well. Remember, animal fats that are solid at room temperature contain mostly saturated fatty acids? Well, the membrane is more like a liquid. So a good share of unsaturated fatty acids essentially gives it this fluid-like appearance. Okay? Now, why do we want proteins? Proteins, first of all, can serve as the transport molecules and also as receptors. They can communicate with other molecules or even cells. Okay? Now, since cell membrane separates the outside and inside of the cell, compounds have to be transported. And this transport is achieved via diffusion, which can be simple or facilitated. It's not a correct term. All diffusion is passive transport, or active transport. So let's take a look at these processes. So imagine, first of all, look at the membrane. These green balls, are they polar or non-polar? Tell me, please. Polar. But the tails are non-polar. Good? So considering this, most of the membrane, like structurally by the volume, is it polar or non-polar? Compare the thickness of the polar heads to the non-polar tails. Make sense? So your membrane is again like a wolf in sheep's clothes, okay? On the outside and inside it's polar, but the inner layer consists of these fatty acid tails, okay? Which means if molecules are to cross the membrane directly, they have to be nonpolar. And they have to be small. Am I making sense? They can cross the membrane directly. And when we say, when we say they diffuse, you can represent diffusion like this. So they go from high concentration to low concentration. Just like a ball running down the slope. Making sense? Now, if molecules are polar, they can't cross the membrane directly. Am I making sense? They will have to use some kind of a channel, just like a door. So we call it facilitated diffusion. But again, facilitated diffusion doesn't require energy because these molecules also move from their high to low concentration. Now, active transport. Is basically moving against the concentration gradient, upwards from low to high concentration. And this process requires energy. It can be in the form of ATP, and there are other options which we're not going to talk about. Okay? So that concludes our lipid portion.